And we are recording this for those people who are not able to make it here on time. And this is the third of 9 groundfish seminars that we have running through December 14th. Since we are all doing this seminar remotely, the speaker will use an electronic pointer or be descriptive when indicating specific points on the slides. To help with this format, please mute your audio and turn off your video feed to avoid additional distractions. Also, please keep your questions for the end of the seminar, or if you think you might forget them, type them into the chat box and Liz and I will compile them after the end of the seminar. Before I introduce today's speaker, I want to remind people to join us for next week's speaker, Madison Hall, who has a dual position at the ASFC and Alaska Pacific University, and she will talk about building a cooperative rockfish survey in the Gulf of Alaska. And that will be held at the same time next Tuesday, October 26th. So today's speaker, Carolyn Sanay, completed a master's degree in fish ecology in 2009. After working in river systems for many years, she started as a redfish stock assessment biologist in 2017 for the Department of Fisheries and Oceans. Her job is to assess the quantity of fish available to better understand the biology of redfish and the consequences that they may have on the ecosystem. Her present research aims to provide recommendations on exploitation rates and management measures to regulate fishing in the Gulf of St. Lawrence, which she'll be telling us today about filling in science gaps in response to an unprecedented increase in redfish.
Thanks, Mark. Thanks for the invitation. Thanks for the presentation. So don't worry, it won't be a presentation on myself today, but I thought it would be good to just start by a few words on my background. So I'm Canadian. I come from Canada. I'm born in the province of Quebec, so this province where we talk French on the east side of the country. I did most of my studies in Montreal. So I did my undergrad in animal physiology. Then I kept working throughout a master's degree in fish ecology, where I looked at river fish polymorphism. Then I was lucky enough to get a contract of 6 years with the University of Montreal to work on a project that was called NSERC Hydronet. It was a network of different universities looking at the impacts of hydro dam on river system and to try to mitigate the influence of flow regime changes on fish community. After this contract, I went down south and visited the state for a year, where I worked for the University of South Florida on a little parasite that is really hard to pronounce for Francophone people, schistosomiasis. So a little parasite that we can find in freshwater system that can infect snails and also humans. And in 2017, I had a nice proposition from the government of Canada, so from the Department of Fisheries and Ocean, to come back in my country, in my province, to work in stock assessment. So I've been working with fish for the past 15 years, and it's quite recently that I added a touch of salt to the water system that I'm working on. So as I said, I'm working for the federal government of Canada, more precisely the Department of Fisheries and Ocean. I work at the Institut Maurice Lamontagne that you can see here on the picture. So we're located on the east side of the country, right next to the St. Laurent River. It's a very nice site. So we're right here. On the little red dots. So it's one of the main Francophone research center, where before COVID, we were more than 450 employees. So please be merciful with my accent, or if I'm looking for some words, it's been a while. I didn't practice my English with those travel bans that we're all living. So Fisheries and Ocean has 3 main axes of priority. The first one is to promote prosperous fisheries. The second one is to maintain sustainable ecosystem. And the third one is to ensure safe waterways. So being in stock assessment, I work in between the two first axes where we're trying to balance economy and ecology. And more specifically, I work on these, whoops, sorry for that, on these cute little redfish that you can see on the left-hand side of the slide. In Canada, we have different management units to manage stock. For redfish, we have Unit 1 that you see here in the Gulf of St. Lawrence, and Unit 2 that is here right under Newfoundland. And for redfish, those two units are managed together and considered as one single stock. The redfish fishery supported an important landings starting in the 50s until the 90s. Here you see an historical picture that was taken back in the days, and the fleet were using both bottom and mid-water trawl at the time. So the presentation that I'm going to give you today focus mostly on Unit 1, which is under my responsibility, but the stock is considered, those fish are considered as one stock throughout Unit 1 and 2. We'll get back to that in a second. So here you see a figure showing the landings across year for different NAFO division. So we see that we had 3 important episodes of high landing back in the days. So one in the late 50s, another one in the second half of the 60s, beginning of the 70s, and the last one was in the late 80s. It was a pretty bumpy road before 1995, when a moratorium was put in place to protect redfish, who crashed along with many other groundfish species. So the moratorium was in 1995, and in 1998, an index fishery started. So a little quota of 2,000 ton was put in place to keep an eye on the stock. And as you can see on the lower panel that focused on more recent years, the 2000 index fishery tag was never fully harvested. We track this stock with our DeFO survey. It's a summer survey happening in August. It started in 1984 and it's still on the go. Along the year, different vessels were used, and each and every time we change boat, a comparative mission occurred to make sure that we could standardize all our estimates. Right now, we're using the Telias that you can see here on the top picture. We're fishing with the shrimp net, so a Campbellin 1800 trawl that has really small mesh, so we're capturing a bunch of organisms. It's a multi-species survey, so we're getting small shrimp and huge fish. The trawl has a mouth opening of 17 meter per 5 meter high. We're aiming to do 15 minutes station, trawling at 3 knots. We're using a stratified sampling design. So here on the lower picture, you see a map of our stratum that we're trying to cover. We'll try to do a minimum of 2 sets per station every year. Once the fish is on the boat, we're gonna measure a bunch of variables such as fish length, mass. We're gonna evaluate their sex, and we'll also gather many biological information. All of this will allow us in the end to have indicator by species and size classes. Redfish are still on the moratorium in Unit 1, but we had a great surprise. In 2011, 2012, and 2013, we saw the arrival of massive cohorts of redfish in the Gulf. The last stock assessment was in 2019, and at this time, we estimated that the minimum trawlable biomass was over 4 million metric tons for redfish. So this is a huge number, but I think that what speaks even more, if we're looking at the figure here, you have the redfish estimates in red and the biomass of all the other species combined in black. And I'm reminding you, we're getting a lot of diversity from small fish, small shrimp to large fish. And in 2019, the redfish accounted for 90% of the total captured biomass as compared to 15% between 95 and 2012. So there's a huge shift in the ecosystem where now it's clearly dominated by redfish, which was not the case not so long ago. So the question that we're asking ourselves right now is how can we get ready as a government to harvest all this biomass? How can we avoid the mistake that we may have done in the past to get ready to sustainably exploit this important resource? One of the main challenge with redfish, it's in the Gulf particularly, is that it's a mixed stock. So we have two totally distinct species, Atlantic redfish, also called Sebastes mentella, and the Acadian redfish that we also call fasciatus. So they're definitely two different distinct species, but they really look alike. We have a trick to get species composition, so a percentage of each species in our survey data, but to get a precise identification at the individual level, we really need to go through genetics. So one of the first thing we updated was the genomic structure of these fish across the Atlantic. So we used a fairly new genetic approach called SNPs, so single nucleotide polymorphism, that reiterated the fact that even if they look alike, those two species are really different and that they diverged about 1 million years ago. And that on top of this interspecies differentiation, there's also a lot of within species variability, where we identified 3 ecotypes of Mantella and 5 population of Fasciatus. If we're looking at Mantella, you can see that the genetic signature, it's pretty homogeneous throughout unit 1 and 2, where we're getting those light blue ecotype, which even goes up north, almost up to the Arctic. The situation for fasciatus is a bit different. If we're looking at unit 1 and unit 2, we see that there's 3 distinct population within unit 1 and 2. So these findings were published a few months ago in Evolutionary Application Journal. If ever you want more details on this topic, there's the title of the paper at the bottom of the slide. If we do a little bio 101 of those fish, usually redfish will be described as a demersal species. We'll find the main huge agglomeration for the commercial fishery along the slope of the banks and the deep channel of the Gulf and the Laurentian Channel. So the deeper areas here that you can see on the map, the deeper areas are in darker blue shade. But redfish can have different behavior. Sometimes it's going to be near the bottom. It can do some vertical migration during the night to go up in the water column to feed on zooplankton. Sometimes we're going to see some homogeneous layers on our hydroacoustic system. Other times they'll do some compact, dense school of fish. So it's really 2 species that can have different behavior depending on time of the day and time of the year, and potentially other factors that we don't even know. We also see different habitats for each species, where Mantella, so the Atlantic redfish, is usually found in deeper areas, so where we have more than 250 meters of water depth, whereas Fasciatus will be found in shallower habitats, So in between 150 to 250 meter. We can have some overlap between both species, but Mantella definitely prefers deeper habitat than Fasciatus. Each and every year, we collect some content during our survey to try to understand who is eating whom. Redfish can be challenging in terms of stomach content because they're physicless fish, meaning that their swim bladder is not connected to the pharynx. So when it's brought up quickly from the bottom of the ocean to the surface, the swim bladder can expand, pushing on the stomach, and part of their meal can be regurgitated. So that's a limit of stomach content, but I mean, it still gives us a good idea of the potential interaction of predation that redfish can have with other species, and also to identify potential competitors to move towards an ecosystem approach. So the main ontogenic shift that we're going to see in redfish, both species in that case, is that when they're small, so under 20 centimeters, they'll mostly feed on zooplankton. As you can see here, some copepods on the left side of the slide. When they get to 20 centimeter around, they'll start including shrimp in their diet. So northern shrimp, which is an important commercial species in Canada, but also a bunch of other species of shrimp that fishermen are not that much interested in. And when redfish will reach the size of 25 centimeters, they'll start eating fish as well. And they can even eat their young. We saw many cases where we found some small redfish in the belly of larger redfish. So there's maybe some feedback in the population, auto-regulation of adults potentially feeding on their own young, sorry. One of the particularity of redfish is that they're really slow growing. But the other side of the coin is that they can live for a very long time. So their lifespan can be around 40 years old, but there's some evidence that they can reach 75 years old if they're not fished before. That comes with a slow growth. So here you see what we think should be their growth curve based on other cohorts that we've seen in the Gulf in the past. So you see that it's fairly slow. In 2019, at the last stock assessment, the size was getting close to the minimum regulatory size of 22 centimeter. It's the minimum regularity size, but frankly, most fishermen are ready to wait a little bit for those fish to grow so that they get to a size that has a larger market values. And if everything goes well, I'm not done validating all our data from the last summer survey, but in 2021, these fish of the 2011 cohort should arrive to a model size of about 25 centimeter. So we need to be patient. It takes time, but the good point with this is that it allows us some time to be prepared and fill the gaps that are missing. Another particularity of redfish that they're ovoviviparous, meaning that they will do internal fertilization with this little external organ that we can see on the male here. So the copulation is known to happen in fall, so in between September and December. The female will carry the embryo in her abdomen throughout the winter, and larva extrusion will happen in spring, so around April to July. On the right-hand side, you see some larvae here. So when they're extruded, they're about the size of a grain of rice. They're more buoyant than the water in the Gulf. So they will go in the surface layer. So at the surface, they'll stay there for a little while and they'll feed, they'll get bigger. and after a few months, they'll migrate down the different stratum, different water strata in the Gulf to reach the bottom. These reproduction events are leading to sporadic recruitment in redfish. So it can take between 5 to 12 years and sometimes even 30 years between strong recruitment events. Here you see the recruitment dynamic, our best call on recruitment dynamic for Mantella in blue and for Fasciatus in red. Please note the different axis on the different value on the y-axis. So Mantella is really doing some crazy recruitment burst, whereas Fasciatus, for Fasciatus, we see recruitment events that are smaller but more frequent. So we can see here in 2011 that we had these massive cohorts, and we're not totally sure exactly why. Is it because the water got warmer? Is it because the timing between the extrusion of larvae and their prey was just perfect? It's really hard to pinpoint the factors that promoted these strong cohorts, in the sense that they're super rare and that we cannot do a lot of stats with a handful of events. But what we know is that we have never seen such a huge recruitment from for Mantella in the past. On the right side, you see the size of redfish that we get, the smaller redfish that we can get in our nets. And when the department saw all those baby redfish coming in, they started in 2016 a management strategy evaluation. So a management strategy evaluation, often called MSC, is a collaborative process where different people, so the federal government, DeFO, but also provincial government, fishermen, First Nation, NGOs, we're all sitting down around the same table to try to develop a management strategy evaluation that would work for redfish. Sorry. We went down that route because in the past, nobody was able to develop one single assessment model to describe those two redfish species, surely because of all the uncertainties we have for these species. So during the MSC, we actually developed 18 different models addressing the main sources of uncertainties. So we had like a lot of what if model. What if we don't have any important recruitment for 25 years? What if their growth rate is reduced by a factor of 2? What if our species composition is wrong and that finally the species specific trends are not exactly what we think they were? So that was a lot of work. And we came up with 21 different management procedures that were evaluated to determine if they met the 7 objectives that I've set all together. So we had some conservation objective. So let's say to have both species reaching and staying in the healthy zone, but also some objectives that were developed by the fishery industry. So having high tax, high quotas, but that would remain stable for as long as possible. So in the end, we had 4 different management procedure that passed all our tests. And it suggested that as soon as 2026, these 2 species could together support harvest level of 40 to 80 kilotons per year for both unit 1 and 2. So that means that soon enough, it could be one of the largest fishery in Canada in terms of volume. So this work that took almost a year and a half was past peer review and was both published within our internal government publication system, but also in CGFAS, so primary journal. You have both title here if ever you want to read more about it. During this management strategy, we were also able to develop reference points. And based on the 2019 estimates, Mantella that we see here was way above the upper reference points, something like 6.5 times, the estimate, sorry, is a 6.5 times larger than the upper stock reference points. So it's comfortably in the healthy zone, but fasciatus was still in the cautious zone. So pretty much halfway between the lower reference point that you can see here with the red line and the upper stock reference points that you can see in both figures, illustrated by the green line. So during the MSC process, fasciatus being less abundant than Mentella kind of act like a choke species. Given that we don't get species-specific estimate of landings, we really have to be careful on not setting the TAC too high, because given that we don't know which species is getting harvested, it could deeply impact fasciatus, which we still have to protect because it's not yet in the healthy zone. So fasciatus was acting as a choke species, and therefore a consensus could not be obtained. And the Department of Fisheries and Ocean didn't move forward implementing any specific management procedures. I won't get into the weeds of those meetings, but we published this year an article in Marine Policy describing the main challenge that we had. So to make sure that the process is really transparent, to make sure that the science team has strong communication skills to explain some models that can be hard to understand for some of the stakeholders. It's also important to define roles clearly ahead of time so that everybody knows what we're expecting from them. And another point that we identified is to have a strong commitment from everyone. So sure, the science team, but also fishermen and managers, because when participants are always changing from one meeting to the next, it can be difficult to keep track of the progress. So in the end, the management strategy evaluation was not officially implemented. The fishermen requested an experimental fishery, which was authorized by the minister. And the experimental fishery had 4 specific objectives. The first one was to try to target Mentella in the fishery. So we developed a new project on this. I'm going to talk about it in a second. The second objective was to minimize the catches of small redfish, for which there is no market, not a high value for fishermen, so we want to leave them in the water. The third objective was to minimize bycatch, because even if it's only a small percentage, when the fishery will be at the level of 40 to 80 kiloton per year, a small percentage can soon become an issue for some species. And lastly, we wanted to have a better grasp on the spatial temporal distribution of both redfish species, small fish, and bycatch. So it's not exactly what the science team was expecting from the MSC, but in the end, it was some pretty cool goals that helped us define the question and the research that we've been conducting since then. So as I said, one of the main gaps that we had is that we didn't have landings at the species level. During our survey, what we're doing is that we're counting the number of soft anal fin rays that you're seeing here in the picture. And by doing so, we cannot identify individual fish to their species, but we can get an estimate of species composition. So we know if in the catch we had a mix of 50-50 Mentella fasciatus, or if, one species was clearly dominating the catch at, like, let's say 80%, 20%. So it's not as precise as what we could have with genetic information, but it allows to cover a lot of fish, a lot of catch at a fair price. So this method that is used in surveys since 2010 is now being implemented in the fishery. So we've developed new protocols, new forms. We train observers at sea, so independent people that are on the boat to take these measures. We created a little clip on YouTube explaining them how to do it. Because a few years ago, it was pretty easy to go visit them and offer training in person. But with COVID, obviously, that was not possible. So we had to change gear a little. We also carried out a bunch of validation in our lab where the sample that were measured on the boat was sent to us, and we recounted all of those annual fin recounts. And we realized that training is still mandatory. Here you see a histogram showing the minimum observation rate between what was done on the boat and what we've got in our lab. And in most of the case, we were under 20% error, but we really hope to get that percentage lower before being able to use this in an efficient way to monitor the fishery. But slowly but surely, I'm sure we'll get there. There's also many collaboration with the industry and university to test different gears to limit bycatch and the catch of small fish. So in the past year, we had many trials at sea looking at different mesh sides, T90 mesh, which is kind of just turning the fabric of the net around. It leaves the bigger openings for run fish, such as redfish, to escape through the mesh. There's also a bunch of folks trying different grids that would allow both small and big fish to escape the nets. Back in the days, so in the 80s and the 90s, midwater trawlers were fairly common in the Gulf, but the knowledge of midwater trawl kind of got lost since the moratorium. So there's a lot of effort to bring that back. So here you see one paper that was published recently, but there's many other ones coming. But it's really not just a matter of publication. Here we have a great opportunity for fishermen to master fishing techniques that would be more eco-friendly. And obviously, it's also a great window for the government to implement appropriate management measures to make sure that we can harvest the fish in a sustainable manner. We're also looking carefully at some specific bycatch species of concern. For instance, here you see the results, some results of scientific advice that we published this year that looked at how we can protect white hake. So white hake is a species that is classified as endangered based on the Committee of the Status of Endangered Wildlife in Canada, so COSEWIC. And those two species, right, those three species actually, white hake and our redfish complex of two species, they really overlap. So here you see the big orange bubbles or redfish and the little bluish dots in the middle that are way smaller are white hake. So it's two species that are really overlapping in terms of distribution. And we did some modeling exercise that showed that we should limit bycatch of white hake to 150 ton if we want to have a good probability to slow down the rate of decline of this species. And we're still looking carefully at if we can find some different tricks, so different months or maybe different gear, different depths that would allow us to reduce the ratio of white hake on redfish to try to, sure, have a nice exploitation of redfish, but without harming other species around. So nowadays, redfish are kind of part of many other stock assessments. If we think one of their main prey or turbot that we think is competitioning with redfish. Redfish is now having a place in many other stock assessment as a key component of the ecosystem. We also updated some key model life history parameters these recent years, especially the maturity ogive. So in 2018 and 2019, over 700 fish were collected. We kept the gonads that were all read with histology to determine reproduction stages. There were also all genotype to update the maturity ogive. And what we saw is a fairly big drop in L50 that were estimated to be between 19 to 25 centimeters in the 90s as compared to 15 to 18 centimeter with this 2018-2019 research project. So it's a fairly big drop in L50. We're working on a paper right now. It's hard to know exactly what the mechanism creating this drop, maybe temperature. It has been seen before that when the water gets warmer, fish may reproduce at earlier age or smaller size, or it may also be something that is density dependent with this strong increase of redfish around. But given that these two phenomenon kind of co-occurred in the Gulf, it's hard to really identify precisely the main cause. It's super interesting in terms of science, but for fishermen, it may be not such a good news in the sense that we've seen often that when L50 are dropping, it may come with reduction in growth rate or L50. So this is something that we'll look into in details in the upcoming years to see what will be the repercussion on growth rate and maximum size. We implemented those new ogive in our stock assessment routine starting in 2011, which correspond to the arrival of the first big cohort of redfish, but also to an important temperature increase in the Gulf. And what we can see is that obviously with lower L50, we get higher estimate of spawning stock biomass. So here, you see the same graph that I shown you before with the black dots being the estimate based on the new ogive. And here I added in blue the results that we're getting with the new ogive. So no big change for Mantella. It was in the healthy zone, still in the healthy zone, but even way higher than the upper stock reference point. But for fasciatus, it meant that in 2017 and 2018, when we're using the new ogive, it brings them up to the healthy zone. But then we had a little drop in our estimate. But still, it means that it changed their stock status, and it may have implication for stock management. Another ongoing project that we have right now is to look at the impacts of global warming, so both temperature increase and acidification on redfish in our lab. There's really not much in the literature on redfish physiology, surely because of the challenge to bring them back alive in the lab. Because when we're trawling and getting redfish, usually they're not in good condition. So the first challenge was really to go get them and bring them alive in our lab. And to do so, we actually use divers. that went at 30 to 40 meter depth. It's not there that we're getting the huge agglomeration of redfish, but they're so abundant right now that even in shallower habitats, we can find a few. And we did a little clip on YouTube to explain to everyone, collaborators or the general public, this project. So I'm gonna show you what it's given and take a sip of water during that time.
Redfish are fish that live in the depth of the Gulf of St. Lawrence. They sustained a significant fishery before collapsing in the 1990s. Under moratorium since 1995, redfish populations are now increasing dramatically. Little is known about redfish physiology given the challenges of studying them in the laboratory. This situation is due to the difficulties of bringing them alive to the surface, because redfish are physicalist fish, having a closed swim bladder. This organ is in fact an air pocket located in the fish abdomen, helping its buoyancy. If the fish is brought up to the surface too quickly, which is usually the case with fishing nets, the air expands as the pressure drops and the bladder stretches. At this point, the organs are compressed and the stomach is often pushed into the mouth of the fish, and the bladder may even burst. Redfish brought up to the surface during fishing are almost always damaged. A team from Fisheries and Oceans Canada has tackled the challenge of capturing redfish alive and healthy. To do this, redfish were captured one by divers using dip nets and then transferred into cages. Afterwards, they were raised to the surface over a few days, performing decompression stops, much like divers do. These breaks allowed the redfish to evacuate the excess gas in their swim bladders and allowed scientists to bring them up to the surface in excellent conditions, providing a stock of about 800 redfish at the Maurice Lamontagne Institute. Once in the laboratory, redfish are exposed to different environmental conditions, varying, for example, the temperature, the oxygen, or the acidity of the water.
In the context of climate change, we'd like to know how our redfish will react to the warming and acidification of the oceans. We will subject our redfish to a range of environmental conditions. We have 16 tanks here, 4 lines of 4 tanks each, and each line can have a different temperature. So we will have a range of temperature starting at 3 degrees and reaching 12 degrees. For each line, we have 2 tanks at a pH or level of acidity that corresponds to what we observe in the deep layer of the St. Lawrence estuary right now, and the other 2 tanks have a lower pH, water that's slightly more acidic, representative of what we expect to see in the deep layers of the St. Lawrence estuary in the second half of the century. We will expose the fish to these conditions for 5 months, during which we will measure their rate of food consumption, their growth rate, as well as their survival rate. At the end of 5 months, we will subject the fish to a battery of physiological testing, including measuring their rate of oxygen consumption through respirometry.
These experiments will help in understanding how climate change can affect redfish survival, growth, and food requirements.
And we can obtain this information by placing redfish in a respirometer, which essentially is a glass bottle like this one, in which we can measure the rate at which the oxygen is utilized.
With such data in hand, we will be able to better anticipate the progression of the population and sustainably manage the redfish fishery. This research will also allow us to better understand the impacts that redfish will have on other species from the Gulf of St. Lawrence, particularly on their prey, such as the northern shrimp, which is also found on our plates.
So there we go for some footage of, let me just bring back my PowerPoint. And the sound should be back on my voice. So yes, this little clip was meant to explain the project to collaborators, but also to the grand public. I think that it's good that Canadian knows what we're doing with their taxes. So it was not as pretty maybe as a coral reef that you may have in the south, but I think that the Gulf of St. Lawrence is still pretty beautiful as well. And that these little redfish are way prettier also when they're alive. Um, and actually, I'm pretty excited because this week we're finishing one of the first experiments. They're actually, measuring fish at the moment in the lab. So it will be very interesting to, know what's the effect of temperature and acidification on mortality, growth, metabolism, food consumption, and their tolerance to hypoxia. This project was first brought up because the Gulf is really warming quickly since 2010. And we know that redfish can live a very, very long time. But given that we didn't even know what was their favorite temperature, it was hard to have an idea of how they would face this global warming. Because you see here the blue and green color are representative of 4 Celsius degree, whereas when it's in red, it's 7-8 Celsius degree. And this warming just kept going further and further. So we wanted to check if we think that those redfish will stick along even with all of those changes. There's a lot of investment proposed by fishermen to gear up for the redfish fishery from the government to improve transformation plans. But before investing millions of dollars, we thought that it would be good to just give a look at how they should face global warming. So that's the end of my talk today. I'd like to thank a bunch of collaborators. Obviously, the presentation that I've done today is not based only on my research, but we're really a group of people behind all of this. So many people from the Department of Fisheries and Ocean, obviously, but also folks from university in France, on the West Coast, in Quebec, all the survey crew that, made our survey possible every year. And obviously, thanks to you guys for listening. And I'll be more than happy to answer questions if I can and have your comments. There's maybe some blind spots that we didn't think of. So all your input is more than welcome.
Well, thank you, Caroline, for an excellent presentation. There's a lot of really interesting material in there, and it's so fascinating to see how this sudden increase in abundance has just caused all these research problems for you. We don't have any questions in the chat right now, but there's our first one. People are welcome to unmute themselves and ask questions to Caroline directly, but I'll read this one from the chat first. And the question is, how did the observed population trends for redfish in Canadian waters compare to those in US waters in the Gulf of Maine?
The Gulf of Maine is closer to Unit 3. If I go back to the map, it's gonna be maybe easier for you to understand. Sorry for flipping those slides quickly. But yeah, there's one stock, the one on which I'm working, and that did a crazy rebound. It's located in Unit 1 and 2. We also have Unit 3 here that has different challenges. When we're going down south, we don't get this mixed species as much. It's going to be mostly fasciatus. So the increase down south is not as strong as what we've seen a bit more up north. I must say that I don't have a lot of collaboration with NOAA, but let's hope that this talk will maybe bring some new opportunities. We're still carrying out a lot of work on, there's a very good genetic lab at the Maurice Lamontagne Institute with Geneviève Parent that has a very good lab, and we're still working on revisiting the mapping of the different ecotypes and population at a large scale. So maybe you guys can help with having some additional sample down south for that kind of project for sure.
Great. And the question was from Sean Rooney, and I apologize, I missed his first question, but I think you answered both his first and second questions, so thank you. Does anyone want to unmute their microphone and ask a question directly to Caroline? Well, while we wait for people, I wondered, we have a lot of redfish populations in the west coast of the U.S. and in off BC and Alaska. And one of the big problems we have is the variability in catches, where we get many catches that are tiny and then a few that are large. Do you have something similar to that?
Sure. When we're looking at our survey estimates, we see that our confidence interval are fairly huge. And this is exactly created by what you're saying, the fact that is really bouncy. You can, get in an area, don't have much redfish, and then move just a little bit, and you see your hydroacoustic system just turning red. And then I think the larger catch that we had on the Telios for 15 minutes, that was like 20 metric ton. So that was fairly fun to sort. But no, that I think it's just part of the redfish biology. That said, we're still tracking them very well. I mean, there's some huge confidence interval, but we're not getting some down sea. We're not getting some bumps in there. Like the trends are fairly smooth. So I'm pretty confident with what we're getting from our bottom trawl estimate. Something that I didn't mention, but that we're working on, it's to try to develop an acoustic estimate for redfish, given that right now they're so abundant that we see them for like many meters up or net. So there's clearly a part of the population that we're not serving as well as we would like to. And this can be tricky because if we're opening a fishery and that the stock is dropping, but that the cloud of redfish is just reducing, but that we don't see it in our net and in our stock assessment estimate, it may mean that when we're finally gonna see a decrease in our estimate, in our indicators, maybe it's too late. So I was not ready to present any result on this right now, but it's something that we're trying to target as well to encompass the whole water column. Obviously, with acoustic, we don't have any information on each species separately. They're pinging the same, but it will surely be complementary to what we obtained from the bottom trawl survey.
Okay, and then there's a question in the chat from Sabrina. She writes, in West Coast rockfishes, we see that reproductive effort is plastic in response to temperature, food availability, and body size. Are you planning any laboratory studies on reproduction in the lab as the fish mature?
It's something that we've discussed with Denny, the researcher that you saw in the clip. The first challenge will be to make them reproduce in the lab. It's not clear. It's something that we're keeping an eye on, and if ever we can really have them reproducing it in our lab, it would be super fun to test for sure. But I'm not so sure that they'll be happy enough in our tanks to exhibit all their natural behavior and reproduce. But that would be super interesting for sure. And it's also like reproduction for ovoviviparous fish is also challenging because there's so much step, right, between having an individual that is mature, but then having a successful recruitment. The copulation needs to work. the maturity of the larva in the female's abdomen as well. They need to come up in the environment when they have the food that they need. Then they need to reach a certain size before getting preyed on. Then they need to migrate down to the bottom of the Gulf of St. Lawrence, which is stratified and has a cold intermediate layer that can be challenging to migrate through. So there's a lot of step between maturity and a successful recruitment event for redfish.
In our trawl surveys, we also have issues with being able to drag our net through all of the habitat types where these types of fish occur. And that's something that Madison, I think, will touch on next week with her seminar. Do you also have problems like that the redfish are occurring in really rough, rocky areas?
But frankly, right now they're so abundant that we see them pretty much everywhere. But yeah, like as in any bottom trawl survey, there's some areas where it's harder to trawl. That said, we're trying to cover as much as possible all habitats, and sometimes we're gonna see the Coast Guard crew repairing our nets more often than they would wish to, but we're trying to cover as much as habitat as possible. Maybe it's less bumpy in the Gulf as compared to Alaska. If we're going back to this map, you know, maybe we'll have some very specific location where we're gonna, you know, go. We're gonna move a bit out of areas that would be super bumpy, but the whole general area is usually covered every year. But that would also be something where the acoustic would be very useful, because it's a tool that, you know, is not impacted by the shape of the bottom.
Okay, we're still not getting anyone who is brave enough to unmute themselves and ask you a question directly. But I was curious to know if your trawl survey on the government ship, do you only work during the daylight hours, or do you also do your fishing at nighttime?
It's both day and night. It's like a little it's a boat that never stops. So we're covering like 12-hour shift and having a shift working by day, a shift working by night. And this is something that is often when we're doing the comparative mission and trying to standardize for a bunch of parameters, testing day and night effect is part of the things that we're looking at. Right now, the signal was not strong enough to suggest that we had to, but we're always looking at if day and night has some effect, because there's some evidence in the literature, scarce, but there's some that suggest, and a lot of the knowledge of fishermen that suggest that during the day the fish would be lower in the water column and that they migrate up in the water column during the night to feed on zooplankton. So if that would be the case, then bottom trawl would be more efficient during the day as compared to night. That said, it's the same thing pretty much every year. Half of the sets being done by day, the other half being done by night. So at least if there's a day and night effect, it's the same throughout the whole time series.
Okay.
But we're gonna change boat again. Hopefully, if everything works nicely, next summer we'll be in comparative mission again, and we'll revisit all the standardization of the time series, and day and night will be included for sure. But will be tested. We'll see if it works including it.
Well, thank you, Caroline. And last chance for anyone who wants to talk to her directly. And there's a typed question in the chat from Ryan Morse asking, where does most of the catch end up? Does it go to export markets, local seafood, fish meal?
This is not a science question, but right now the fish are still small, so the landings are not super high. If I'm correct, most of it goes to Europe. It's kind of a nice white fillet that kind of competes with tilapia, which is also super cheap. So there's a lot of work being done by the industry to develop some new market. It could be bait when it's too small, could be round fish for Asia. I heard that is something that is super popular to have a whole redfish in your plate. In Asia, it's supposed to be promising. Back in the days, it was a lot. The main exportation before the moratorium was actually the state for a lot of fish and chip in the militaries. So we'll
Interesting.
see who's gonna buy them this time. I'm not sure, but I'm sure that the industry will find some market for these nice, tasty, clean redfish from Canada.
Last minute question.
Hey, Mark, this is Wayne Paulson.
Wayne, thanks for joining.
Just a comment, really. When you look at the Aleutian Island and Gulf of Alaska stocks of Pacific Ocean perch, we've seen a similar dramatic increase over time in their abundance. And your talk really reminds me of that. The increases that we've seen, though, have led your increases by many years. But you might take a look at the stock assessments for those two species and see if there's any comparative problems that have been addressed on this side of the continent compared to yours.
Will do. Thanks for the heads up.
Yeah.
Thanks, Wayne. Reached the top of the hour, so we should probably cut it off there. If anyone wants to speak one-on-one, perhaps after we stop the clock, maybe Carolyn, you can stay around for a few more minutes just in case.